Hi, my name is Jayra Highland, and I'm going to be talking about the gut microbiome and multiple myeloma. To start, let's talk about why we care. The gut microbiome is a critical player in human health and disease, influencing immune responses, drug metabolism, and cancer progression. In the context of multiple myeloma, where treatment responses vary widely among patients, understanding the microbiome's role can pave the way for personalized medicine. With the rising prevalence of multiple myeloma, there's an urgent need for innovative strategies to enhance treatment efficacy, reduce resistance, and improve patient outcomes. This study not only addresses this gap but also explores an entirely new perspective by focusing on microbiota-host interactions. So what was being asked? The central question was, how does the composition and function of the gut microbiome influence treatment responses in patients with multiple myeloma? Specifically, the research sought to determine which microbial communities are enriched in patients with favorable treatment outcomes versus those with poor responses. How do these microbes alter drug metabolism or immune modulation pathways, thereby impacting therapeutic efficacy? So what was achieved? The study achieved its goal by demonstrating the specific nitrogen recycling bacteria that are significantly enriched in patients responding well to treatment. It highlighted the microbial metabolic pathways that influence drug efficacy, providing strong evidence of a direct connection between the gut microbiome and treatment outcomes. The contribution made by this study is substantial. It identified potential microbial biomarkers for predicting treatment responses, suggested microbiome-targeted interventions like prebiotics or probiotics as a viable supplement to existing therapies, and it opened doors for further research into microbiome-cancer interactions, setting the stage for precision medicine in oncology. Background. The gut microbiome has emerged as a critical area of study given its significant influence on human health, immune regulation, and drug metabolism. Recent advancements in microbiome research have revealed its role in cancer development, progression, and treatment outcomes. For hematological malignancies, blood cancers, like multiple myeloma, the gut microbiome offers a promising path to understanding the variability in treatment responses among patients. While prior research has linked microbiota to chemotherapy efficacy and toxicity in solid tumors, studies exploring its impact on blood cancers, particularly multiple myeloma, are still in the early stages. The article I've chosen builds on the prior evidence, focusing specifically on microbiota-host interactions and their influence on drug efficacy, which could potentially revolutionize therapeutic treatment strategies in this field. Need. Understanding the role of the gut microbiome in multiple myeloma is both timely and critical. With the rising incidence of this hematological cancer, there's an urgent need for innovative treatment strategies that can improve patient outcomes. Current therapies often vary in efficacy, and patients experience significant differences in how well they respond to treatments. By exploring how specific gut microbes impact drug metabolism and immune responses, our research aims to address these challenges. The broader implication of this work is substantial. Not only do the authors highlight potential biomarkers for predicting treatment responses, but they also open the door to microbiome-targeted therapies. These insights pave the way for more personalized and effective approaches in treating multiple myeloma and potentially other cancers. The article I've chosen is called "Alterations of Gut Microbiome Accelerate Multiple Myeloma Progression by Increasing the Relative Abundances of Nitrogen Recycling Bacteria." So the authors of this study set out to answer a key question, and that's, how does the gut microbiome affect the success of treatments in patients with multiple myeloma? They wanted to explore whether specific gut bacteria play a role in how well patients respond to their cancer treatments. Their findings could help develop personalized therapies based on an individual's gut microbiome, potentially leading to better treatment outcomes for multiple myeloma patients. Getting into the methodology, we're going to talk about data collection first. So They collected 37 stool samples. 19 of the participants were multiple myeloma patients, and 18 were healthy control patients. They also collected a fasting serum sample, and these samples were used for FNT. So the population details of the participants were of similar ages and BMIs, and none of them had gastrointestinal diseases. In case anyone is unaware, FNT stands for fecal microbiome transplantation, and this is a method that's used to do experiments with mice. Sequencing techniques. They use 16S rRNA gene sequencing to identify and classify the bacteria. This specific method targets specific regions in the bacterial DNA to profile microbial communities. They also use shotgun metagenomics. This provides a comprehensive view of all the genetic material in the sample, and this helps us to understand the functional potential and identify genes that could be linked to drug metabolism. So getting into data analysis, on the left side here, I've made a list of the programs and methods used in the study. This is not all encompassing because there were a lot of them. These were simply the ones that I thought were the most important. So on the right, we have metabolomic analysis, and this was used to link metabolites with treatment responses and to identify possible pathways that could be influenced by the microbiome. So getting into the statistical approach, we have some statistical methods that were used to form the paper's conclusions. They primarily used PCOA based on the Bray-Curtis dissimilarity index, which calculates how different two sets of data are by comparing the presence and abundance of a variety of elements such as species or genes. And if there are values from 0 to 1, 0 meaning they're identical and 1 meaning they're completely different. They also used alpha and beta diversity analysis, alpha being diversity within the sample, beta being differences between the samples. And these aid in understanding the diversity and balance of the microbial communities. They also use correlation and regression analysis to identify relationships between microbial abundance and treatment outcomes, and the bacteria with positive versus negative responses to said treatment. So one of the first important results they found was that multiple myeloma enriched bacteria results from accumulated nitrogen. So there are 8 modules: denitrification, dissimilarity, nitrate reduction, transaminase degradation, and you can read the rest because they're very hard to pronounce, but they were mapped and based on identified differential KEGG orthologies, and 3 of them are involved in the nitrogen cycle. So prior research showed these authors that nitrogen levels can allow for modifications in the microbiome, and this led them to believe that dominant multiple myeloma microbes result from the accumulation of host-secreted nitrogen. So here in figure C, we're going to be looking at the growth rate of 2 different treatments compared to a third. So right in the middle there, you can kind of see on the far left side, you have the fastest growing tumors, and you can tell that by the fluorescence in the bodies. And then on the right side, you see significantly less of that fluorescence, which means that the tumor growth is significantly slower. So here we have the author's proposed mechanism of interaction between host and the microbes. We can see the urea coming in from the kidneys and the nitrogen from the bloodstream, which will come together in the gut, and these are used by the nitrogen recycling bacteria to create GLN, which is deposited into the bones, and the bones then put out more nitrogen. This is extremely important because they found with their research that urea and nitrogen buildup in the body cause an increase in malignancy. So moving on to our discussion, how does the gut microbiome influence treatment responses in patients with multiple myeloma? What we know so far is that nitrogen recycling bacteria significantly impacts malignant progression. There's a direct link between the gut microbiome and treatment efficacy. Bacteria can act as biomarkers for predicting treatment responses, and there's a potential for personalized therapeutic treatments. So the broader implications of this are insight offered paves the way for microbiome-targeted therapies. There's potential to enhance treatment outcomes. It's extremely important to integrate microbiome analysis into clinical practice and to promote a more personalized approach to medical treatments. What we don't know is the precise molecular mechanisms that cause this, whether it's generalizable across other cancer types, and what the long-term effects on patient health and treatment outcomes will be. So a few quick points in conclusion. The gut microbiome is a key regulator in cancer treatment. Integration of a microbiome analysis into clinical practice is where we should be heading in the future, and improvement in patient outcomes is definitely a possibility with these microbiome-based treatments. Now getting into our discussion questions. Number 1, what additional studies are necessary to validate these findings? Number 2, how might the gut microbiome affect resistance to certain drugs? Number 3, are there any ethical considerations that we should account for when manipulating the microbiome for therapy or treatment? Number 4, could changes to a patient's diet improve the effectiveness of their treatment? And number 5, what role does the patient's genetics play in determining the impact of the microbiome? And this brings us to my references slide. Thank you guys so much for watching my presentation. And here are the links to all the articles that I use for my background research and my main article.